Hi, I'm Mark von Zastrow from UCSF.
And I'm Anastasia Henry, a graduate student in Mark's lab.
Today we'd like to tell you about a study that Anastasia has carried out together with James Hislop, Joe Grove, Kurt Thorne, Mark Marsh, and me. And it concerns the regulation of clathrin-dependent endocytosis of a particular signaling receptor by ubiquitination. Many signaling receptors, upon ligand-induced activation and initiation of downstream signaling, are regulated by a variety of mechanisms, including clustering in clathrin-coated pits, which then mediate regulated endocytosis, removal of a receptor from the cell surface. This process of regulated endocytosis can have numerous effects, one of which is to destroy receptors by degradation in the lysosome and effectively downregulate the signal. Another is to return receptors to the cell surface by recycling, and this can sustain signaling and sometimes change the properties of the signal. So the process of clathrin-mediated endocytosis has been studied for many years, and it's well established that it's a precisely choreographed process, regulated at many levels, from initial formation of the clathrin coat to the subsequent endocytic scission and formation of the endocytic vesicle. The mechanisms by which this occurs involve proteins associated with the clathrin coat itself, as well as lipids that are accumulating near the clathrin coat. Generally speaking, endocytic cargoes, including the signaling receptors that we study in the laboratory, have thought to be passive players, essentially inert passengers in this process of endocytosis. The work that Anastasia has done has revealed a level of active control of the process of endocytosis by the signaling receptor itself, by virtue of ubiquitination of the receptor.
So what do we mean by this? We examined the question of cargo control using the mu-opioid receptor as a model, and we noticed an unexpected effect of receptor ubiquitination on the endocytic rate of receptors. This was revealed using a combination of flow cytometry and live cell imaging using turf microscopy, which allows us to selectively follow events happening at the cell surface. We found that when we prevented receptor ubiquitination from occurring, which we did by mutating the receptor's cytoplasmic lysine residues to arginines, the endocytic rate of receptors was greatly reduced. Turf microscopy revealed that when we blocked receptor ubiquitination, the receptors were still able to efficiently enter clathrin-coated pits, but that the later stage of endocytic trafficking was affected. By following the dynamics of individual receptor-containing clathrin-coated pits and scoring their surface lifetime, we found that when we blocked receptor ubiquitination, we significantly increased the amount of time that receptor-containing pits persisted at the cell surface before undergoing endocytic scission. We were also able to identify the specific sites that are required to regulate this process, which happen to be two lysine residues present in the receptor's first intracellular loop. So our findings revealed the existence of a local signaling system that is able to control the dynamics of the clathrin-coated pit by sensing the presence of individual receptors through their ubiquitination. So after receptors become activated and phosphorylated, this leads to the recruitment of the endocytic adapter protein, arrestin. Arrestin can then link receptors to core components of the clathrin machinery, promoting the entry of receptors into clathrin-coated pits. Receptors themselves, we found, are able to actually stall the lifetime of the clathrin-coated pit, effectively engaging a brake and allowing the continued enrichment of receptors to occur. Arrestin itself can then recruit the E3 ubiquitin ligase, SMURF2, to activated receptors, promoting their ubiquitination. So here ubiquitin is acting as a signal to communicate information to the clathrin-coated pit to control its dynamics. This signal is sensed by the endocytic associated protein epsin through interactions with its ubiquitin interacting motifs. Epsin can then basically act as an endocytic brake release, triggering efficient endocytosis. So what are some functional consequences of this kinetic control mechanism that we've identified? Well, one function that we identified is that the lysine residues that are important for controlling the endocytosis of receptors are precisely the same sites that are required for the efficient destruction of receptors via lysosomes. So one possible function of this control mechanism could be to optimize later steps of trafficking in the endocytic pathway, basically acting as a biochemical checkpoint to stall clathrin-coated pits at the cell surface that contain receptors until these receptors have undergone appropriate ubiquitination.
A second consequence of this local signaling mechanism is that it exerts a rather strong effect on receptor loading into the clathrin-coated pit. Although receptor ubiquitination did not affect initial coat formation, nor did it affect the rate at which receptors could accumulate in clathrin-coated pits. Prolonging the surface lifetime of the clathrin coat caused effectively an overfilling of the clathrin-coated pit and increased the variability in the number of receptors present in the coated pit at the time of endocytic excision. So we think another function of this kinetic mechanism is to control the surface density as a counting mechanism, essentially, to control the surface density of receptors in the endocytic membrane.
So to wrap things up, while endocytosis can clearly regulate the function of signaling receptors, our work suggests that the converse is also true, that signaling receptors themselves can precisely and locally regulate the endocytic process.